participants to kindly mute yourself, please.
Karthik sir, can you stop sharing, please?
Stop sharing, sir.
Yes, sir.
You can now. This is can operate.
Sure.
Confirm the communication recording.
Yes, ma'am.
செல்லு பார்க்கணுமா அது வெபினார் அட்டெண்ட் பண்ணிட்டு இருக்கேன். ஒழுங்கு
உங்க சாரிக்குடா
ಹೌದಾ 5 ಸಿಕ್ಕಿದೆ.
Miss Kanishka, could you please mute yourself? Miss Kanishka,
Yes, ma'am.
yeah, please mute yourself, ma.
நகலை எடுத்து போனது என்ன இந்த நகலை எடுத்து हाँ तब चलो
Hello. Am I audible? Please confirm.
Yes sir.
Yes, sir.
Thank you. Thank you.
Sir.
Good morning to all the dignitaries, respected
Good morning,
resource
sir.
person, faculty members, resource scholars, and dear participants. I request all the participants to kindly mute yourself. I am Dr. Mohan Gandhi Bontu, Associate Professor, Department of Pharmaceutical Analysis, and one of the coordinator of CAPS along with Dr. Babu, Associate Professor. On behalf of CAPS, Center of Advanced Analytical and Bioanalytical Sciences, Department of Pharmaceutical Analysis, JSS College of Pharmacy, UT, it gives immense pleasure to welcome you all to today's webinar on advanced bioanalytical method development using LC-MS industrial practices, challenges, and regulatory perspectives. We begin by expressing our humble pranams to His Holiness Swamiji for his divine blessings. We express our sincere gratitude to the management of JSS Academy of Higher Education and Research, Mysuru, for their constant vision and unwavering support. It is because of their strong commitment to quality education, research excellence, and industry collaboration that programs like this are made possible. JSS College of Pharmacy, UT, proudly holds the 4th rank in India as per NIRF 2025. And this achievement reflects the strong academic foundation laid by our management. We are truly grateful for their continuous encouragement. We are deeply thankful to our respected principal, Dr. S.P. Dhanpal sir, for consistently motivating students and faculty to go beyond textbooks and engage with industry-oriented learning. His continuous support has played a crucial role in the successful planning and execution of this webinar. We also extend our sincere thanks to the head of department, Dr. N. Krishnaveni ma'am, for her constant guidance and leadership, which keeps our department active, progressive, and academically vibrant. I am delighted to share that this webinar has received an overwhelming response with around 1,560 registrations from 18 different states across India. Among them, nearly 1,000 participants are students and research scholars, and over 500 are faculty members. This clearly reflects the enthusiasm and growing interest in advanced bioanalytical sciences. In the current pharmaceutical landscape, bioanalytical techniques, especially LC-MS, plays a pivotal role in drug development, pharmacokinetic studies, and regulatory compliance. The increasing demand for accurate, sensitive, and robust analytical methods highlights the importance of continuous learning in this domain. This webinar is organized with the objective of bridging the gap between academic knowledge and industry expectations, and to provide insights into advanced bioanalytical method development practices. We are fully honored to have our esteemed resource person with us today, whose expertise will undoubtedly provide valuable knowledge and practical perspectives. I extend my heartfelt gratitude to all the participants for your enthusiastic presence and active engagement. With this note, I once again welcome you all and wish you a highly informative and engaging session. Before we proceed, I would like to invite our HOD ma'am and in-charge principal, Dr. N. Krishnaveni, to give her valuable thoughts on this webinar. I also request her to give brief glimpses about the Department of Pharmaceutical Analysis. Over to ma'am.
Thank you, sir. Submitting my humble pranams to the lotus feet of His Holiness Jagadguru, Shri Desigendra Mahaswamiji of Suttur Mutt. A warm good morning to the distinguished speaker, Dr. R. Karthik, the faculty members from the Department of Pharmaceutical Analysis, the research scholars, as well as the faculty members who have joined as participants from various parts of India, and the enthusiastic student participants. I'm here to give a brief overview about JSS College of Pharmacy as well as the Department of Pharmaceutical Analysis at JSS College of Pharmacy, OT. So JSS College of Pharmacy is one of the premier institution which is spread around 6.7 acres of land specifically for pharmacy program. At JSS College of Pharmacy, we are having programs right from D Pharmacy, that is Diploma in Pharmacy. We also offer an undergraduate program, B Pharmacy, a PharmD program, as well as M Pharm in about 10 specializations. And in order to enhance the multidisciplinary research activities, we also provide for the UG students as well as for the PG students. We also organize postgraduate diploma programs as well as certificate programs in the relevant specializations of pharmacy, as well as JSS Academy of Higher Education is also organizing various programs in life sciences and medicines, for which our students will also be getting the access. In addition, within the campus, we are now having around 170 research scholars who are pursuing their PhD program. And JSS College of Pharmacy, Ooty, is ranked 4th in India by NIR of 2025. We are an affiliated college to JSS Academy of Higher Education and Research, Mysuru, and JSS Academy of Higher Education and Research, Mysuru is NAC A++ accredited. In addition, JSS College of Pharmacy, Ooty, is also certified with ISO 14001:2015 and ISO 50000:2018, both for environmental management system as well as the energy management systems. In addition to the regular academic activities, the college, JSS College of Pharmacy, is also having few specialized centers like DST approved TIFAC core for carrying out the research in herbal drug. And we have a government approved drug testing laboratory specifically for Ayush related activities. And we also have a drug testing laboratory which was established in the year 1994 under the aegis of the Department of Pharmaceutical Analysis, which is which is approved by the state government of Tamil Nadu, and it is also NABL accredited. So we cater the needs of not only the pharma students related to education, but also consultancy activities for industry, developing industry academia relationship through Center for Advanced Drug Research and Testing, which is called as CADRATS. So this is a brief overview about JSS College of Pharmacy, Ooty. And in regards to the Department of Pharmaceutical Analysis was established in the year 1994 with an aim to establish a PG program in the pharmaceutical analysis. So M.Pharm Pharmaceutical Analysis was established in the year 1994, and subsequently M.Pharm in Pharmaceutical Quality Assurance was established in the year 2010. And the department is also recognized for registration in PhD programs right from 1994. And presently we are 4 research guides who are capable of carrying out the research activities in the department. The department has got 3 well-established laboratory. One laboratory is earmarked for high-end instrumentations, which caters the needs of the research activities pertinent to the college as well as to the department. The research facility comprises of an liquid chromatography with a tandem mass spectrometer by Shimadzu, which is containing a triple quadrupole mass spectrometer. We do have a liquid chromatography with a single quadrupole mass spectrometer and an array of HPLC instruments with various detector configurations like UV detector, UV visible detector, photodiode array detector, refractive index detectors. And in addition, we do have a semi-preparative HPLC instrument with a PDA detector. And in addition to these sophisticated instruments, we do have the regular instruments which are used for academic purposes like UV visible spectrophotometer, Fourier transform infrared spectrophotometer, fluorimeters, conductivity meters, pH meters, and so on. So the basic research activities in the department includes analytical, bioanalytical method validation, especially for pharmaceuticals, herbal formulations, and for chiral formulations. So we deal with all the types of the formulations, analytical, bioanalytical method validation. Then we are also working on the stability studies, that is development and validation of stability indicating analytical methods for pharmaceuticals and herbal compounds, along with establishing the forced degradation profile of these formulations, plus identifying the impurities and characterizing the structure of those impurities. And the third research area we are presently developing now is moving towards the development of point of care devices for most of the societal problems. And we also work on the nutrikinetic assessment of functional foods. So these are the specific research areas of the department. And as I have already mentioned, through the drug testing laboratory, which was approved by the state government of Tamil Nadu and NABL accredited, we cater the needs of the society, especially we act as a third party testing agency for some of the pharmaceutical industries, along with some Tamil Nadu Medical Services corporations. Thank you.
Thank you, ma'am, for your wishes and briefing about the department. Now, I request the participants to allow me to present few things about our recently established Center of Excellence, that is CAP. Hope my screen is visible to everyone. Once again, welcome to all the participants. So this is the first program. This webinar is the first program which we are organizing under the aegis of Center of Advanced Analytical and Bioanalytical Sciences, abbreviated as CABS, which comes under the Department of Pharmaceutical Analysis, JSS College of Pharmacy, Ooty, with the main motto of bridging analytical excellence and industry-ready skills. The vision of CABS is to establish and serve the research fraternity in general and pharmaceutical analysis in particular by providing basic analytical and bioanalytical services to advanced analytical and bioanalytical solutions. We also try to develop skilled human resources in the same domain. Why CAPS? We have been seeing, or we have been witnessing in present pharmaceutical sector that there is high demand for high quality analytical data. So where do we get high quality analytical data? Only from the high end analytical equipment like HPLCs, UPLCs, LCMS, and LCMSMS. Unless we provide high quality analytical data, regulatory agencies like ICH, USFDA, EMU, etc. are not going to accept your reports. This is the one end. And the other end is the same pharmaceutical industry or sector is requiring skilled professionals, especially in advanced instrumentation. So this is the gap what we generally found, because the people who are coming out of the academic organization, though they have theoretical knowledge, they do have a gap in accessing or handling high-end analytical equipment. So CAP is going to address this gap by acting as dual platform. One is we are going to provide all the analytical services, right from basic analytical services to high-end analytical services. More than that, we are ready to train all the people who are having the interest in learning analytical equipment. So this is one solution platform for both analytical services as well as training building. The core strength of the center lies on state-of-art instrumentation. As ma'am has already said, we have equipment right from basic UV visible spectrophotometer to high-end LCMSMS. We do have LCMSMS, LCMS, and 4 HPLCs of both manual injections as well as auto injections. We do have complementary analytical equipment like atomic absorption spectrophotometer, FTIR spectrophotometer, UV visible spectrophotometer, flame photometer, photofluorimeter, basic digital pH meters and conductivity meters, along with nephlotherapy. We do have sample preparation and support systems. We have our own water purification systems, dissolution apparatus, solid phase extractions, ultrasonicators, and other equipment. What I mean to say here is we have each and every equipment related to analytical field, right from basics to the advanced equipments. The key activity what this center is going to cater or do is we have been dividing, or we are divided the key activities into 3 pillars. The first pillar is service oriented. The services what we are providing here is the basic method development. You can develop your method right from basic UV visible spectrophotometer to high-end LCMSMS. You can validate your method. We can perform pharmacokinetic studies. We can perform bioanalytical and bioequivalent studies along with stability or force degradation studies. We do conduct impurity profiling and characterization. We are ready to go even with analytical QBD methods along with greenness techniques also. So these are the services what we are going to provide under this particular pillar. And the second pillar and the main pillar what we are going to have here is the FTPs. We are conducting or we are ready to conduct all the faculty development programs which can enhance analytical skills among the faculty members. We are ready to conduct workshops as well as hands-on trainings in whatever numbers you want. All these workshops and hands-on trainings are tailor-made. You can come and visit our center in a number of 3 to as much as 20 or 25. So all our programs are tailor-made. What you want, you just inform us. We can have the programs designed exclusively for you. We are ready to provide certification courses on current burning topics related to analytical and bioanalytical techniques. We do provide skill development courses for the people who are really interested to enter into industries. Along with that, the third pillar is the research. Research is mainly focusing on collaboration with academia as well as industries. The industries can be small scale industries to the large scale industries, and the academic collaborations will be or can be from a small college even to the universities. We are mainly focusing on the translational research, and we are ready to go even with the interdisciplinary research also. We are not restricting ourselves to only pharmaceuticals. We are even ready to take the challenges of Ayush products also. So these are the key activities what we are going to have. So if you feel that our center is good for you, you can contact any of these numbers. Babu, Dr. Babu, Associate Professor, and myself are the coordinators, or you can contact any member of the department for your information. And the only thing what we are asking or what we are requiring from your side is only interest. If you have interest to learn, if you have interest to dedicate yourself for practicing the high-end equipment, you are good to go. Thank you very much for your time. And the second program, what we are going to start in the month of May.
I'll explain.
Yes, ma'am.
Yeah. So, good morning to all the participants once again. The Department of Pharmaceutical Analysis is conducting a 3 days workshop, especially a hands-on workshop, which is sponsored by the Indian Council for Medical Research, Department of Health Research, New Delhi. It is entitled Computational Pharmacokinetics and Virtual Bioequivalence Using Artificial Intelligence and PBPK Modeling, that is physiologically based pharmacokinetic modeling tools for translational research. The hands-on workshop is organized from 14th to 16th of May 2026. It is not a hybrid program. It is an offline program. It will be organized at JSS College of Pharmacy, OT. So the benefits of the program, if you see, you would be able to have hands-on experience on some of the biopharmaceutics tools like PK Sim, then bioequivalence study tool like Winonlin, that is Phoenix Winonlin software, as well as the biopharmaceutics tool GastroPlus. So the program is for 3 days. All the 3 days, morning session will be the lecture and afternoon session will be the hands-on training module. So can you please zoom this? Next page, sir. Before previous page.
This one.
Third page, yeah. So this program, who can attend this program? Any academician, faculty, research scholars, or undergraduate students who are in their final year, or PharmD students who are from 3rd year to 6th year, or 1st and 2nd MPharm students of any specializations can attend this workshop. Clinical pharmacologists or industry professionals who are working in drug development and formulation, and or bioinformaticians or computational biologists who are interested to attend can attend this program. The registration for the workshop is limited only to 100 participants, and and the registration fees is 1,200 rupees inclusive of GST. And for this registration fee, we will be providing breakfast, refreshments, as well as lunch on all the 3 days, along with a workshop kit. And for this particular tailor-made program, we have got eminent resource persons, especially from academia as well as from industries. We have Dr. Rajkumar Malayandi as an eminent speaker who will be giving more insights into the GastroPlus software. He will be teaching us how to utilize the GastroPlus software as well as how to handle that GastroPlus software for all our biopharmaceutical purposes. He is professor and head at the Department of Pharmaceutics, Naipur Hajipur, and to handle the Phoenix WinOnline software for giving a practical demonstration as well as to talk about the applications of WinOnline softwares. We have identified 2 scientists who will be from various industries, like Dr. M. Sirajuddin. He is the principal biostatistician at SDG Clinicals Private Limited, Hyderabad, and Dr. Sundaramurthy Nainar. He is the director of PKPD division, Endopharma Inc. So these are the 2 experts who will be handling regarding the Phoenix WinOnline software, a demonstration on Phoenix WinOnline software. We also have another eminent speaker, Dr. Chandra Teja, who is a biopharmaceutic specialist from Dr. Reddy's Laboratories. He will be giving us insight into the hands-on experience for PK Sim solutions. In addition, we also have Dr. K. Shashanka Prasad, the faculty from JSS Academy of Higher Education and Research, who will provide insights on doing ADMET characterization for the newly developed molecules, or screening the ADMET properties of the various molecules which are developed, and the implications of the application of AI on the regulatory requirements. There will be a lecture by Mr. Gopal, manager from Global Regulatory Affairs, Pfizer Healthcare India Private Limited. So we have identified the experts, especially from industry as well as from academia. And the registration is limited only to 100 participants. The last date for the registration is 30th of this month. So if anybody who is interested to participate in the particular workshop, you can contact any of our department faculty, and the registration link is available in the brochure. And for the benefit of the participants, the brochure will also be shared in the CAPS WhatsApp group. Thank you. And in addition, I also take the privilege in thanking our eminent speaker for today, Dr. Karthik R. Dr. Karthik R has got a vast industrial experience, and he will be able to provide valuable insights into the practical workflows, problem-solving strategies, and compliance frameworks which are followed globally, especially for bioanalytical method development and validation. So I request all the participants to actively engage, interact, and make the most of the session as a learning opportunity because they are vital for enhancing your knowledge, fostering your innovation, and preparing yourselves for the dynamic demands of the pharmaceutical industry. I also place on record my sincere appreciations to the organizing team, especially Dr. Mohan Gandhi Bontu, Associate Professor from our department, and the coordinator, one of the coordinator, Dr. B. Babu of the CAPS, for all their efforts in bringing together this meaningful academic interaction. I also take this opportunity to thank the leadership of JSS Academy of Higher Education and Research, Mysuru, as well as the principal, Dr. S.P. Dhanapal, JSS College of Pharmacy, Ooty, Vice Principal, Dr. K.P. Arun, JSS College of Pharmacy, Ooty, for providing an opportunity for establishing such spirit hubs. So we have got sanctioned 7 spirit hubs. Each spirit hubs are unique in their own way, and one such spirit hub is the Center for Advanced Analytical and Bioanalytical Sciences, which will be functioning under the Department of Pharmaceutical Analysis. And for establishing the spirit hub, we have also received the financial assistance from JSS Academy of Higher Education and Research. So I take this opportunity to thank all the leadership for making this possible, and this is one of the first event. I wish the CAPS for organizing such many events successfully. So I wish the webinar a grand success and hope it will be highly informative and inspiring for all. Thank you.
Thank you, ma'am. Now I would like Sangamitra ma'am, Assistant Professor, Department of Pharmaceutical Analysis, to kindly introduce today's resource person.
A very good morning to one present here. Myself, Dr. Sangamitra, Assistant Professor in the Department of Pharmaceutical Analysis, JSS College of Pharmacy, Ooty. On behalf of Center of Advanced Analytical and Bioanalytical Sciences, JSS College of Pharmacy, Ooty, I extend a most cordial and respectful welcome to each one of you to this online webinar organized under our Industry Academia Interaction Lecture Series. So with a deep sense of honor and professional admiration that I introduce to you our distinguished chief guest for today's webinar, Dr. Karthik R., Head Bioanalytical and Analytical Divisions, Scitus Pharma Services Private Limited, Chennai. So he's a highly accomplished pharmaceutical scientist with over the 2 decades of dedicated expertise in bioanalytical and analytical sciences. So he holds a Doctor of Philosophy in Pharmaceutical Sciences from JSS Academy of Higher Education and Research, Mysore, in the year of 2023. So on his current capacity as Head of Bioanalytical and Analytical Division at Scitus Pharma Services, so he provides a strategic and technical leadership to a team of 59 scientists and then operating 13 LC-MS systems and 1 ICP-MS instrument. He oversees bioanalytical method development and validation for small molecules and peptides, as well as the analytical work encompassing the nitrosamine impurities, xenotoxic impurities, and elemental analysis in drug substances and their products. So in full compliance with international regulatory standards. So on this professional record is a benchmark of excellence. So he has a successfully developed and validated method for over 500 molecules, and he has managed more than 700 regulatory studies submitted to the US FDA and then MHRA and then EMA agencies. So he was the architect of Scitus Pharma bioanalytical facilities, which achieved and then accreditations under the ISO and then regulatory approval from the US FDA. So beyond his industry contributions, he also an active contributor to pharmaceutical sciences. He is the author of over 15 peer-reviewed publications, and also he serves as peer reviewer for international reputed publications, including drug designs, development, and then therapies. So today, Dr. Karthik will share his invaluable expertise on the subject advanced bioanalytical method development using LC-MS. So in industrial practices, challenges, and then regulatory perspectives. So this is a topic of critical relevance to students, researchers, faculty, and the industry professionals, and we are privileged to receive this knowledge from a practitioner of his category. So your presence today is both an inspiration and an honor of our institution for every participant of this webinar. So we thank you most sincerely of your time, your wisdom, and your unwavering commitment to advance the standards of pharmaceutical sciences. So here I extending a warm and respectful welcome to our distinguished chief guest, Dr. Karthik R. We welcome you, sir.
Thank you. Thank you. So first, I thank you. Thanks, Department of Pharmaceutical Chemistry of JSS College of Ooty, HOD Dr. Krishnamani Madam, and the coordinator for CAPS, Dr. Mohan Gandhi, and Dr. Babu, for organizing this training for the students as well as for the faculties and from throughout the India. And I congratulate for the CAPS to be get succeeded, and what they have today, they have been started. They have to achieve a lot of things in future, and they have to do a lot of things to the pharmaceutical peoples and to the students to engage more and to gain more knowledges from the CAPS. And I will start my today's presentation, and it's good morning to all. And today's presentation is advanced bioanalytical method development in LCMS, MS. It's an industrial practices, how we will be doing it, and what are the few case studies, is what challenges we have been faced during the method development, and how we have been overcome those things. And the regulatory perspectives is for the bioanalytical, what the recent FDA's queries has been come, and how we can able to resolve it, and those things we'll be seeing in today's presentation. So first, what is a bioanalysis? it's a subdiscipline of analytical chemistry. It covers the quantitative measurements of the drug and the metabolites in the biological fluids like blood, plasma, serum, and urine. So it starts from 1970s onwards. The most of the universities or the researchers are from the companies. They start identifying the PK or PD related and what is the metabolism active diseases from the biological matrices. They used to start, but earlier then when they do the analysis, they used to do with the HPLCs and the methods. What they do it's somewhat the tedious process and time consumption will be more, and the cost also it will be increases. So they use a lot of solvents, and the timing it will be taking more than 6 to 7 hours to process, and to get the results it will be takes more than a day also. So from there it starts, and then there were advances in the techniques starts improves, so that everything it is been slowly it has been getting changed. And now the in 2020s, so entirely the advancements, everything it has been changed, and mostly we have been using LCMS, MS, either the HPLC or UPLC will be used based upon the companies or the institutions where they have. So these advanced techniques makes everything simple, and advanced in the extractions procedures and advancements in the column chemistries. So these all makes easy, and make the time consumptions also very less, and to develop the reliable data. Now it has been the still from 1970s to now also the regulatory perspectives also been increased more, and their expectations is also it's increased a lot. So whatever they expect is what we deliver the results or the data to the regulatory. So it should be perfect, and it has to be reliable. So that is the main thing which the regulatory it is expect. So for the reliable data, whatever we want to do it, so may they expect it has to come from the LCMS MS because of its advanced technique. So the bioanalysis which bridges the chemistry, biology, and medicine. So by that we are delivering a reliable data for the drug development. And the objective of bioanalysis is to provide a quantitative measurements of the active drug and its metabolites. So especially for pharmacokinetics, where ADME profiling can be done, and pharmacodynamics, the target and engagement and responses we can able to identify by through bioanalysis, and toxicokinetic studies. So how much exposures of the toxicity level of the particular drug it can be able to identify by bioanalysis, and bioavailability. So how much fraction of the drugs it reaches the circulations, that it can be understand by this bioanalytical technique, and bioequivalency. So the equivalency between the innovator drug and the generic drugs, and confidence intervals, and all this it can be able to identify by using these bioanalytical techniques. So the bioanalytical methods, apart from this, again, therapeutic drug monitoring purposes also we have been using it. And this quantitative bioanalysis delivers a decision and critical data across the entire drug development continuum. So this is very important for any drug development, and this particular analysis is highly important for understanding the nature of the drug, how it behaves in our body. And this, when it comes to this one, it's a LCMS MS. So this technique what we are going to see here. So the LC, it's high performance liquid chromatography, either that or it can be an ultra performance liquid chromatography. It's been coupled with mass spectrometry. So why we use this is it's this integrated technique. It's more selective, sensitive, and quantitative rigor. So where this selectivity it comes from the HPLC or from the UPLC. So where it can able to separate the compounds in a complex matrix, where MS is based upon the mass by charge ratio. So the particular only the particular compound of light of interest it can able to be get quantified. And still if we are having a multiple components or a fixed dose combinations, or a drug with different multiple metabolites, everything it can able to get easily quantified by using the mass by charge ratios, because each mass it will be an unique, and if same mass if it is there also, the fragment ion which we are going to get it, that it can be we can make it as a different, and we can able to quantify from the fragment ion. So that is the MS technique we are going to use it. So with these both together, HPLC and MS, it ensures an unambiguous identifications. And when we come to the sensitivity, if you see the MS detections, which enables the quantitations of the sub-picogram per ml levels also, and where with the low noise background and the measurement what we get it from this output from this mass spectrometry is at trace level analytics also we can able to measure, and the data which we generated through the mass will be reliable, and the combined platforms which delivers a dynamic range of less than 4 order of magnitude, where the FDA and ICH guidelines as criteria as everything it can be met if you have been using this LCMS technique. And this is a gold standard platform for regulatory grade bioanalysis in modern drug development. So coming to the liquid systems, that is liquid chromatography systems. So the LC part, it contains different components. The pump. So the pump which generates a flow of the elementos from solvents to the reservoirs to the system, and it goes to the mobile phase will be pumped and then it will move to the injector. So this injector is used to inject the based upon the sampling loops, and the samples will be introduced through the flow of element to the column. So the column is a separations is performed inside the column. So based upon the analyte of the interests, we are different column chemistries are there. So we'll go through in upcoming slides also the how to select the columns of those things also. And it has a degasser. So degassers uses a special polymer membrane tubing to remove the gases in the mobile phase, and column heater. So this helps for the LC separation. It's influenced by usually by the column temperature. It will be keeping us a 40 degrees, so it can work from 30 to 60 degree temperature. It can work. So based upon the separation we want, so we can change the column temperature and we can increase the separations of the mixtures. And this ensures a robustness, reproducible chromatographic integrity. So across the diverse biological matrix. And this liquid chromatography, when we come to know that there are two different mobile phases will be there. So one is reversed phase and another is normal phase chromatography. So in normal phase chromatography, the stationary phase is a polar packing. And the mobile phase will be a non-polar, and this applications is for water sensitive compound and for geometric isomers and chiral separations. This normal phase will be used, but mostly this normal phase is for absorption driven separation will be done by using the normal phase chromatography. Most industries, they have been using the reverse phase chromatographic techniques, where the stationary phase will be a non-polar packing. For example, like C18, C8 column, phenyl, biphenyl, phenyl hexyl, cyano, RPamide. So these are the different chemistries of the stationary phases it can be used. And this reverse phase chromatography, you can use it for all type of analytes like polar analyte, non-polar, ionizable molecule. If it is ionic, so ionic species also it can be used. And for this reverse chromatography, the mobile phase will be polar across organic. The water will be the polar, and then organic will be the methanol and acetonitrile. Is the only two organics will be used for this LCMS volatility. And the buffers which we have been going to add in the aqueous, it can be a formic acid, acetic acid, and trifluoroacetic acid, ammonium acetate, ammonium formate, or ammonium bicarbonate. It's in either of these based upon the pH where we work on based upon analyte pH. So either of these volatile buffers can be selected. So only these buffers, the limited only if this is because the MSS works based upon the ionization, and it's for evaporations it has to be done. So these are the buffers which are volatile, so it can be easily get ionized and it can be vaporized and it can enter inside the mass spec. So apart from this, others which generally we will be using like phosphate buffers or other things as for HPLC cannot be used for an MS instrumentations. And moreover here the pH also is controlled. So based upon the analyte pKa, so we have to control the pH of the buffers which we are using for the mobile phase. So the retention increases with analyte hydrophobicity, and this reverse phase works based upon the partition driven separation. And here the pH also is roles a major thing. the depends upon the pH, the ion suppressions it can be happens in the MS. So the pH selection in the mobile phase plays a vital role for here also. So understanding the phase of polarity logistic is a foundational to the method development. So which type of polarity and which one mobile phase we are going to select it, right, and how the pH we are going to select, and then based upon that, the ion suppressions, how it is going to happen in our chromatography. So everything it will be done based upon the selections of these mobile phases. So when we come for the mobile phase selection and consideration, and the strategical selections of robust retention time, peak shape, and ionizations all depends upon the mobile phase how we will be selecting. So as already I told, the organic solvents, why we are using acetonitrile and methanol is that, so because they are very low viscosity, acetonitrile is lower in viscosity, and it will give a sharp peak and higher UV cutoff. So whereas methanol is a strong element strength, the cost effective, and it's a more compatibility with the MS. And these both are volatile. So these both are the component organic solvents which are compatible with for the MS. And aqueous components as already told, and these are the components so will be used for the as a buffer. So in pH adjustments, so we have to see how we are going to adjust the pH. So the pH adjustments all depends upon the analyte pKa value. So before when we start the go for the method development, so we should understand the physiochemical properties of the analyte. And from that analyte, especially we have to know so whether it is an hydrophilic or lipophilic in nature, and what is the pK of the particular analyte, and based upon the pK of the analyte, so how we are going to ionize the analyte by using the mobile phase has to be judged based upon the pK of it. So the mobile phase, we have to adjust the pH 1.5 units from the pKa value. Suppose if you are selecting the analyte is an acidic form, then we have to retain longer in column means then we have to change the pH lesser, that is 1.5 unit of the pKa value of analyte. And if it is a base analyte, so we have to retain it for longer means then we have to adjust the pKa value plus 1.5. So these two things is it has to be taken care, and based upon that pKa value on which we are going to adjust, so we have to select the buffers. So if it has been acidic, so we have used a formic acid because its pK is 3.75 and TFA pK 0.23. So based upon that, which we are going to adjust, that it has to be selected. And for base also same. So ammonium acetate pH will be 4.8, ammonium formate pH 3.8, and ammonia still if it goes to pH 8, 9 like that it will come. So ammonium bicarbonate still it's 8 to 9 the pH it will be there. So based upon the analyte which we are going to do, so that particular pH it has to be adjusted in the mobile phase buffer, and that we have to use it. So by adjusting this one, the volatile buffer selecting properly, the sensitivity in mass it will be get increased, and we can able to avoid the adduct formations like ammonia adduct or sodium or potassium adducts will be avoided when we select these volatile buffers. And next is the separation. So when we selected the mobile phase, so how we are going to do? So either it's isocratic separation we are going to do, or the gradient separation. So if it is in a single molecule, and then we can go with an isocratic separation, it may be is good. So isocratic separation is the mixture of mobile phase is consistent over the complete testing time. So the single combined mixture, it's example, it's 65 to 35%, 65 organic and 35 is the aqueous. Entire the runtime, total runtime, it remains the same. So the limitations for using this isocratic separations is that if two different things to be get separated, if two analytes are there, then one if it is eluting late and one it is eluting faster. So in such cases, isocratic separations may not be work out. So in that such cases, we have to go for gradient separations. So gradient separations, we can start the polarity with aqueous more, and then we can increase slowly the organic phase. So when we come to the end, the organic phase will be get increased. So in such way, if you do it, the mobile phase strength changes on continuous basis. Either we can change mobile phase ratio or we can change the flow rate also. So either way, if you do, it's both comes under the gradient separation. In these cases, if we have a different multiple analytes or different natures of the analytes, if it is there, we can able to separate it by using this polarity. We can able to separate the analytes earlier, so where we can able to retain it properly. The selectivity will be there, and the resolution between the peaks will be more when we select these gradient separations. If it is in a single analyte, then if you can go with an isocratic separations. If it is a multiple analyte with the different eluting patterns, as if it is there, then we can go with the gradient separations. So where we can able to achieve the good retention time, resolutions, and selectivity also will be more when we select for the gradient separation. And next comes the mass spectrometer. So in mass spectrometer. So coming to mass spectrometer, so these are the different components are there: ionizer, accelerator, deflector, and detector. So the ionizers where from the column whatever we are getting it will be get ionized in these particular places, and these and as a stream of vapor it will be get comes over here, and there only particular ions is what we have been there, so that it will be goes to the accelerator. So accelerator where we mentioned the mass by charge, what we mentioned. So only those things will be the running. So MS is a triple part. So first MS is only the parent ion. So whatever the molecular weight we enter, only that particular molecular weight will be entering over there, and that remaining all others will be get deflected by the voltages what we will be applying it, and through the vacuum turbo vacuums which they have been built in the mass spectrometer. So through with the vacuum, it will move towards the next quadrupole, the second quadrupole. So where the fragmentations will be taking, and from the parent, the different fragments will be produced. So there we have to mention which particular fragment it has to enter inside the deflector or to the detector. So that we have to mention, and from there other fragments will be get deflected by the voltages what we apply it, and only the fragment ion of the particular parent ion will move to the detector. So from the detector where the ion to the current conversions and digital MBS quantifications will be taken, and the output will be getting as a chromatogram. So this is where the different components of the MS it is there. And before that, I have been mentioned the column. So the selections of the columns. So based upon the hydrophobic, if it is an hydrophobic compound, so we can select C18, C8 columns as it can be selected. If the compound is a polar compound, and if it is have a strong dipole or in the structure, if it is there, then we can go for a cyano biphenyl phenyl hexyl columns. And suppose the polar compounds, if it is having a phenolic group or acidic group or hydroxylated or hydrogen bonding, if it is been more, so then we can select a RP amide column. So these are the column selections based upon the nature of the analyte which we have been selected. And coming to the sample preparation. So sample preparations is so three different techniques as we have been using for the general technique. We have been using it for extractions of the samples from the plasma or different matrix, either plasma, blood, urine, or serum. So in this, the three technique, the first one will be the protein precipitation technique. So the protein precipitation Nick. So before that, we have to see that whether the analyte which in plasma, so what we are going to get, if it is a non-ionized form or in ionized form, first we have to see the plasma. So before extraction, so we have to see which state that particular ion it is been there. So if it is an acidic compound, the compound should be non-ionized to ensure efficient binding to the reverse phased sorbents. So we have to dilute with the acidic aqueous buffer like 0.1% formic acid, right? And we have to make it in a non-ionized form. So for extraction purposes, if it is an acidic pKa, if it is there, then we have to make it as a -2 of the pKa value by adjusting the pH to -2. So pKa -2, we have to do it, and that particular buffer it has to be selected. Suppose if it is a base compound we are going to do, so what we have to do it, so we have to dilute the specified buffer like ammonium hydroxide, sodium hydroxide, right, or sodium bicarbonate, sodium hydrogen phosphate, or potassium hydrogen phosphate. See, these are the few buffers for basic sites we can be used, and we have to make it as in the neutral form. So the basic compounds in plasma, the pH it has to be in +2 units. We have to adjust it. So by adjusting these pKa values based upon the pKa values, by adjusting these, we can able to easily make the compounds in non-ionized form in the plasma, so which we can able to easily extract it, and the recovery of this based upon the acidifying or basifying the plasma, the recovery will get increased or decreased. So this recovery to increase it is based upon the nature of the compound and how we are going to adjust the pH in the plasma. So once if these particular sections, if it is been taken care, then the extraction techniques is what I've been told. So 3 different extraction techniques. The first one will be the protein precipitations. Usually protein precipitations, if the protein binding and for the particular drug, if it is more than 95 or 98 like that, if it is there, the high chances are there that particular analyte it can become through the protein precipitation extraction we can create it. The protein precipitations usually will be done by acid precipitation or alcohol precipitation. For acid precipitations, we use a perchloric acid, trichloroacetic acid, etc. And what it will be doing it will protonate the carboxylic group, and net positive charges will get lost, and solubility will get collapsed, and the drug will be get released in the acid that we have been transferred. And in alcohol precipitation, if you see, we'll be adding an organic solvent. So like acetonitrile and methanol regularly will be these two only the organic solvents we are using it for alcohol precipitation. So this, which what it will be doing it, this alcohol will be reducing the aqueous environment polarity in the plasma, and it exposes the hydrophobic domains and aggregations of proteins will be taken care, and the drug which binded in the protein will be get released in the organic solvent which we have been added. At this protein precipitation technique, the loss will be minimal. The expensive of the reagents which we use will be less, and the time processing consumption for this particular extraction time will be very low. But whereas the chances are there for significant ion suppression and due to the availability of the phospholipids. So there is a lack of robustness also, possibilities it will be there when we go with this technique. And advancements of this particular technique is the solid liquid extractions. What it is there, it's like a solid phase extraction, similar to that it will be there. So once after doing this one, that particular the final solution, it can be passed through the cartridge, and where the phospholipids chances which makes this ion suppression in this technique can be removed. So SLE assisted cartridge method. So once after completion of this, we can pour through the SLE method where the activation only is required, and other things for that particular SP methods may not required. These phospholipids will be binded, so only analyte molecule will come, and these ion suppression can be eliminated. After this, if you use it, solid supported liquid extractions method. So here how usually will work is that sample additions of 100 microliter to 200 microliter will be plasma will be added in microcentrifuge tube, and the precipitating agent will be added, either salt also will be using it, like zinc sulfate can be used, or ammonium sulfate will be used to salting out the technique, and acid or alcohol. So anything which we have been doing it as precipitant agent, so that will be added, and it will be centrifuged at 4000 RPM or 13,000 G for 10 minutes at 4 degrees centigrade. So the plasma will be formed as a pellet, and the drug which is there in the plasma will be moved towards the precipitant agent, and that we can able to remove as a supernatant. And based upon that, either it can be evaporated and then reconstituted and it can be injected, or we can dilute it with the buffer and directly we can go for an injection. And this, it has to be checked. There is no any ion suppression. Direct dilution also it can be done. So the entire method, its time consumption will be less, but chances are ion suppression will be there. To avoid that ion suppression, and SLE cartridges can be used to avoid this. And next is the liquid extraction. So the liquid extraction is based upon the partition coefficients. So where the analyte will be transferred between the two invisible phases. So our plasma will be one phase, it will be there, and the organic which we are going to add will be the other phase of the solvent, it will be there. So the separation process consisting of transfer of solute from one solvent to the other solvent. So the plasma which contains the solute is the analyte of interest. So when we add the organic phase, so organic phase like organic solvents, we'll be using MTB, DCM, diethyl ether, ethyl acetate, N-hexane, N-heptane, chloroform, or in some cases a mixture of either of the solvents with some IPS also can be added. If the polarity of the drug is more polar, then IPA can be added with this organic solvent, and that the quantity also of this particular organic solvent should be more than that the plasma matrix. Usually, if we are going to take 100 microliter of plasma matrix, if it is there, then at least we should have 1 to 1.5 ml of the organic solvent has to be used, and we have to mix it, and that much volume is required to move the solute, that is the drug, from the plasma to the organic phase. And that particular organic phase separately will be removed it, and that we are being evaporating, and will be for that only the volatile organic solvents only we have been selecting, because once it has been solute has been moved towards the organic solvent, it has to be supernatant to be removed, and then it has to be evaporated, and for that it should be in volatile nature. So this is advantages. It's a short method development time. Cost also it is low. Oh, and only the disadvantage will be the, see, we will be wasting an organic solvents. Sometimes if you are using different proportions, and chances of emulsion formation will be there. Once out of centrifuges, the plasma will be get floated also, chances will be there if it is not been done properly. So only this is the disadvantage it is there, but most of the majority of the methods will go with this liquid extraction technique. And next is the solid phase extractions. It's a very targeted form of sample preparation. So it allows to isolate your analyte interest while removing any interference compound that which have been presented in your sample. So there is a different chemistries are there in this solid phase extraction technique. So C18, strong cationic exchange, strong anionic exchange, weak anionic exchange, and mixed mode cartridges. So different chemistries of SP cartridges been available. So based upon the compound and the polarity and pK of that one. So usually the SP methods which is having the very low pK value or in minus pKa, if it is there, then most of those drugs you have to go with the solid phase extractions. And if any liquid extractions or protein precipitation extractions, if we get a matrix ion suppression or enhancement, if it is there, then obviously we have to switch towards this solid phase liquid extraction. And here the workflow, it will be a loading of the samples is before loading, that particular cartridges has to be get conditioned to activate the pores which is there in the bottom of the cartridge. Once if it has been get activated, then we have to load the sample which has the analyte. And because the plasma samples, it won't have only the analyte, it may have the phospholipid, endogenous metabolites, protein fragments, everything will be there. So we have to hold other things, or we have to wash it out other things, and we have to dilute only our analyte of interest. So the next step will comes the washing step. So there we have to wash the polar lipids or other matrix components and phospholipids, is whatever it is there, it has to be get washed. So for that washing step we have to do. So we have to either use a organic the 5% or 10% organic with like 5% methanol or 5% in water, it can be used. And if you select a different chemistry, based upon that, we can go with an 100% methanol also, or either any other organic solvents like DCM, ethyl acetate can also be used to wash it. Because those organics that depend on the chemistry, so it will be get binded. If you use organic, it may not come down. But if we are going with an C18 or hydrophilic lipophilic cartridges, so methanol or acetonitrile, if it is added, so it will, the pore will be get activated and the analyte will be get diluted. So in cases 100% organic cannot be used in the wash step. And once if it has been washed, where we have been removing all the polar lipids and other matrices components from the cartridges, and the final will be the elution step. So we can go with either 100% organic solvent, or if we have the mobile phase with some ratios around 80 to 70% with the organic and aqueous. So we can add directly the mobile phase also as an elution solvent. It can be eluted, and that eluent can be directly transferred in the autocellular vial, and it can be injected in the instrument. So if we are going with 100% organic solvent, then we have to go for an evaporating that eluted sample, and then we have to reconstitute and we have to inject in the instrument. And this is a highly selective for the method, and this method is highly effectively to remove the phospholipids and any endogenous interferences. Yes, and in this advanced technique will be there are micro elution plates were there. So indeed, instead of using an individual cartridges, the micro elution plates where the volume of the solvents is everything will be required in microliter level only. So 100 to, when we use as a single cartridges, we may require 1 ml or 2 ml organic solvents in each and every steps. Whereas if you go switch over to the micro elution plate, so there we require only 100 to 200 microliter of organic solvents will be required, and the plasma requirement also will be very less, and the usage of organic solvents can be reduced if micro elution plates have been used in rather than these individual cartridges. The casting will be get reduced, and the timing also will be get reduced. We are moving towards the switching towards the micro elution plate. And the costliest thing of this out of these three methods is this solid phase extraction, because the cartridge price itself it will be costing more rather than using the reagents. So this is costliest method. So the preferences will given be a list for this is when the LLE methods or the protein precipitation methods are not suitable, or if the pKa value for the analyte is in highly polar, it's negative if it is there, then these solid phase extraction techniques will be selected. If any matrix affectation if this comes, then this solid phase extraction technique will be selected for the method development. So this is the key takeaway from these extraction techniques is how we'll be doing that end-to-end bioanalytical workflow. So the sample collections will be done, and then it has to be preconditioned with the pH adjustment in the plasma or the matrix what we are using it, and then extraction technique will be doing it. that it will be injected to the analytical instrumentations and there we'll be getting at the final output. So this is the end-to-end workflow integrations for the bioanalytical workflow. And the few case studies I have been picked, 3 case studies is the method development, what it has been done, and which stages we felt some problems is how rectifications we have been done. So each method's 3 cases have been taken. So one case is ion suppression in plasma. So for that, the solution is that post-column infusion and IS normalization is the method solution. And one more case will be a selection of pseudo ion MRM. So pseudo ion MRM is the parent ion and the fragment ions has been remain the same. And so because of that, few problems as we have been raised, and what changes is the solutions we have made it a product ion selected, and column changes also we have been done. And one more case is a degradation of analyte in plasma. So because of that, initial stage we didn't face any problem. So when it goes to the stability, then we faced a lot of failures it is there. And then the stabilization of a plasma prior to processing has been done. So we stopped the degradation. We'll see the cases one by one. The first case is the quantification of dexamphetamine and the L-amphetamine, the dextro levo form of the amphetamine in the human plasma by UPLC and mass spectrometry detection. So this is a chiral separation, and usually for chiral separations, we will be having challenges is in separating this chiral compounds itself. So apart from this chiral separations, we had other challenges also, but during method development, we didn't face anything, and still this is a controlled substance. And first, this is the initial conditions which we have been used for the quantification of this DLL amphetamine. So the column we have used is Lux 3 micron column. It's 150 mm 4.6, and from phenomenons, and this is especially this particular column is only they have do it for the amphetamine separations, it will be there. So they have told this particular column for amphetamine, there will be good resolutions will become. So we used this particular column, and the mobile phase condition is a methanol 2 millimolar ammonium acetate with the pH 11 and 70:30 ratio and 20 microliter injection volume. And we did it for 8 minutes run time. And we did method development, pre-method validation. So we didn't face anything. So all the validation parameters as per ICH-M10 guideline we have been done. So method validation is completed, and when we move to the sample analysis, so we face the issue that the analyte and IS response gets varies irrespective of the concentration. There are some decrease some places, and some places that is increasing. So there is the matrix ion suppression or enhancement, it has been observed. Whereas the response ratio, because we had internal standard, so that it has been taken care, and the response ratios remain same. The concentration what we obtained, there is no issue. But when we see as a IS response, there is a increase, sudden decrease, is there is no constant response. We didn't observe it, and we thought that at the adverse mobile phase component, which affects the ionization consistency. So what the data what we have received from this, it's reliable, but as there is a differences between the samples, ups and downs is there. So the regulatory will not be accept this one. So what we went for, do the redevelopment of this one by changing the column infinity lab portion 120 chiral with 2.1 micron and particle size 2.7 micron. So diameter 2.1 and particle size 2.7 micron. And here what we did, we removed entire aqueous from the mobile phase. So our mobile phase remains as a methanol, acetic acid and ammonia with flow rate 0.25 ml per minute, and we have reduced the injection volume as well as runtime also we have reduced it. So when compared with both the things, so we did changes in the column chromatography. So once we revised this one, so this is the chromatogram, the first method, how we got it, the resolutions between the both is more. it's almost 0.8 minute. It is there, the resolution between the D and L. But when we go with the clinical sample analysis, there was an ion suppressions between the samples as we observed. Where in method 2, when we changed it, the previous method is 8 minutes runtime, the second method is a 5 minutes runtime. And the previous method, the first eluting will be the L-amphetamine and second eluting will be the D-amphetamine. When we changed it, the chromatographic conditions with the mobile phase, yes, D-amphetamine comes first and the L-amphetamine comes the second, and the resolution is 0.4 minutes only it is there. The resolution, even though the resolution little bit less is there, but the reproducibility of the results and there is no ion suppressions has been there, and 37% runtime has been reduced and throughput has been increased, and throughout the clinical sample analysis, the consistency was there. So method wise, both robustness was there, but consistency in the response is not observed in the method 1, whereas in method 2, the improved response consistency is been observed, and the runtime also we have been reduced. The throughput also has been increased. So this is the differences which we have made and how the improvement it has been done. So the method 2, 37.5% faster throughput has been increased, and the cost also it has been reduced because of the runtime and the injection volume, and the mobile phase flow rate also have been reduced. So these are consumptions of the solvents and manpower, everything has been reduced. This is the case 1, and the case 2 is the quantification of triantine and N1 acetyl triethylamine, the triantine and its metabolite in human plasma by UPLC-MS spectrometric detection. So this the triantine usually it's a low sensitive molecule. So as such, if you detect as a triantine, so we may not get sensitive results. So for that, what it has been done, we have been done and derivatization by using FMOC. So that derivatized for both drug and metabolite only we have been used for analysis. So the method 1, and this is the condition. The column we have used is C8 column we have used, and the mobile phase 0.1% formic acid and acetonitrile and 10 millimolar ammonium formate. We have gone for the gradient program. And this we have used pseudo MRM. That is the same parent ion only, it has been used for the fragmented ion. So because of this same pseudo ion we have kept, we got an interference peaks in internal standard of triantine D5, as well as in the metabolite we got an interference peaks. To avoid that particular interference peak coming to the next injection, carrying of that particular injection in next run, so we have increased the runtime to 15 minutes. So this 15 minutes runtime with this particular thing, it has been worked, and method development and validations we have been done. There is no issues and all. So, but we faced a lot of cost involvement in this one, because the runtime is 15 minutes and the throughput will be very less. And but this we did it clinical studies also, there is no differences from the sample analysis, or there is no differences in the variations or ion suppressions, nothing is there. But still, when we got more studies to be done with this one, and with this 15 minutes, it can be more. So what we did, we changed the method. We thought of changing it by selecting the fragment ion for that parent ion, and we thought to change the column also. So the column C8 it is there. This is the second chromatographic condition. So we used the high purity advanced columns. So C8 column with amide polar amide also it is there in this particular column. So we thought of going with still amide with polar, and we'll can check it out like that. We used this mobile phase ammonium 0.01% ammonium hydroxide in 5 millimolar ammonium formate with acetonitrate 75:25, and fluoride 0.8 ml. We have made a split, 1:1 split, and injection volume we kept 5 ml, 5 microliter. So with this, what it has been done, the entire run time from 15 minutes, it has been changed to 3 minutes. So we didn't get any additional peaks with this, and almost 5 times the runtime has been reduced. So 80% of the throughput has been improved. So the main reason is redesigning of the MRM and eliminating the pseudo ions by changing it to the fragment. One more fragment we have kept it, and that carryover interferences with the additional peak is resolved. And polar amide column we have been selected, so which resolved the shortened retention time, and the derivatization part, there is no changes there. So only the column and the mobile phase we have been changed, and the total chromatography conditions is it has been changed to 5 times it has been reduced and increased the throughput. And with this also validated, and we have done studies. So both the studies, the results remain the same, but the way what we have been throughput what we got it is entirely different. So where 15 days running studies is, here it can be completed in 2 days. So where we selecting in that particular column and the mobile phase as well as the ions what we are going to do it, there also it plays a vital role of throughputs can be increased or reduced, it can be done. So these selections also it has to be taken when we have been doing a development. And these are the differences it is there. So changing the method, 80% faster analysis has been happened. And the last case, it is in the diethyl propione. So in this case, what it's also an narcotic drug. It's a antipsychotic drug. So diethyl propione, and key observation what we have been observed during the method development. So PA precision and accuracies and re-injection stabilities. So those things, everything it has been doing during the initial development. We didn't face anything. So there is a reproducible of the results that have been observed. So in method development, when we do it, the matrix stabilities like bench star stability and free star or prepared sample stabilities, auto sample stabilities, we observed QC's failure. So there we have observed that there is a degradation pattern of the analyte over the period of time it has been done. So we did few changes like keeping in ice water bath or keeping it in yellow light conditions and tried it, and these have not been worked out. So then we tried it with the stabilizing of the plasma before extraction. So the stabilization strategy, what we have used it, 5% or 5.5 molar ascorbic acid has been added to the plasma prior to processing or prior to spiking. So this stabilized the plasma, so which makes the preventing the oxidative degradations of diethyl propione. These results degradations of analyte in plasma has been completely stopped. So no degradation products have been observed, and the stabilities are meet correctly as per the ICS guideline. So this is the method one accuracy where fresh is there is no accuracies are made, whereas the stabilities which few I have been listed out here all are having it has been less than 85%, whereas benchtop stability we kept it for 24 hours. It has been reduced to 70%. Stability has been entirely reduced. So after changing the method by using the stabilization of the plasma, so there is no differences. After that, we keep it in the bench also 24 hours also. There is no degradations of the analyte as observed, and the combined status for the stabilities also has met. So the common challenge patterns is in method one is all related to the mobile phase and column chemistry and sample stabilization. So these three parts is only we have to go through it when we go for the method development and when we see any issues as in chromatographic conditions. When these three changes, if we do it, there the runtime it can be reduced, our method robustness will be increased and cost will be reduced, throughput will be increased, and we'll be getting the data which is reliable and the regulatory acceptable data will be produced, and that it will be give more or confidence to the regulatory on the data what we will submitting it. And next things is that FDA from the key learnings is that so early identifications before if you go for a clinical samples, if any early identifications if it has been observed, then the timely optimization can be done. And non-aqueous mobile phase can significantly improve the ionization consistency for chiral separations. So that it has been we had a problem by using an aqueous in the mobile phase. And column chemistry selections also directly impacts on selectivity retention time and overall runtime efficiency also. So when we do it, try method developments, so we should check with the different chemistry columns selections also, even though if we get it, when we do the trial in initial, if we get good resolutions or good thing, and we have to try with other chemistry columns also to see still whether we are going to get a better runtime, lesser runtime, and to give high throughput. So that also has to be checked, and don't go blindly with the single first one what we got it. It's not to stick on to that particular chromatographic conditions. So we can try, even though if we have a good condition, we can try it still whether we get the better conditions than that which we have been got earlier. So that it will make a high throughput for the analysis purpose it will be there. And sample stabilization strategies is also has to be considered because some of the analytes are liable in the biological matrices. So to identify what type of stabilization to be done, either in this case it's an ascorbic acid, but other cases it may not work out for the based upon the type of degradations it happens is in the biological matrix. The stabilization agents is it has to be selected and it has to be checked whether it has been stable or not. And method optimization, the analytical performance requirement will be done. So these optimizations balances will increase the operational efficiency, and the documentations also for these whatever we got the problems is and whatever we do in development, and each cases it has to be properly documented. So it will give knowledges for the future development projects in any such cases if it has been occurred. So that it will be giving a good knowledge that earlier same cases we have been faced some issues. So for that particular cases, this is the solution we have been provided. So that second time when we face such a problem, no need for wasting the time, and same solution which we have been implemented earlier, it can be tried it and then we can sort out the things. And coming to the regulatory aspects, so there are 8 key FDA 483 observations areas. So 483 is nothing but the observations form where the US FDA they will be providing to the companies. That particular form is the 483. So the key points as observation areas is where the regulatory look into it is this I have listed out, and we'll be seeing one by one what is the individually what they look into that and how we can resolve all those things. So data integrity audit trial, method validation gaps, ISR deficiency, run acceptance rejections, study director or QA unit oversight, SOP and training failures, sample accountability, and document change control. So this is the 8 areas where the major observations are occurred. Some cases warning letters is also been issued based upon the point which they have been given to the CRO or the facility. The first one is this data integrity and the audit trial. So the FDA findings is that missing or incomplete raw data files. So either missing of review signatures or missing of the preparer sign or incomplete files is placed in the file. And in instrument, if you see there is a manual deletion or overwriting of the original records, and unreviewed or unsecured electronic audit trails, paper and electronic records contradicting each other, backdated or reconstructed entries, CRO substituted failed BE study with reanalyzed data to pass BE limit. So they did initial analysis and they have submit when they saw that results, the BA data it has been failing. So what they did, they reanalyzed and then they showed that it is passed. So this triggered that, and recent 2024 or for one of the CRO, they have issued a warning letter for doing reanalyzing the failed data and making it as pass. So to avoid these expectations is for missing details is everything. So we have to follow ALCO plus principles, like documentation should be legible, contemporaneous. We have to keep all original and the accurate data has to be reflected. And when you come to the audit trials, all the instruments audit trial should be enabled, and no delete option should be there in the instruments, and audit trial reasons and electronic signatures has to be provided, and no sharing of the passwords should be done. And whatever we go for the reintegration, and original chromatograms also should be there, and the reintegrated version also should be there, and both it has to be submitted to the regulatory, and no deletions of raw data should be done. If anything, it has been a problem has been occurred, so we have to annotate and then proper justification for the it has to be mentioned, but deletion should not be happen. And the hybrid, if it has been there, the paper and electronic, both if it is there, both reconciliation and it should be properly reconciled and it should be controlled, and removing or deleting or discarding of this, it should not be happen. So all data actions traceable to a named individual. So electronic signature, if it has been there, so it is responsible of the particular person who do it. For them, it will be there. So in such a way, we can avoid this FDF finding. And next one is the limitations to the method for this. This is selectivity. So there are multiple fixed dose combination drugs will be there. Fixed dose combination drugs with the different methods, or in some cases the drug will be dosed, and to avoid any adverse event, we will be dosing some concomitant medications during the clinical stage. So in such cases, some of the CROs, they have been and quantified only the drug, and they didn't fail to prove that concomitant medications how far it is going to have an impact with the main analyte. So that it has to be done. So that it was not been done. So we have to demonstrate that selectivity with 6 different lots, including on hemolyzed and lipemic, and matrix effect ion suppression engagement, and not characterized across the donor lots. So this matrix effect also, it has to be done with the 6 different lots, including hemolyzed and lipemic. And when it comes to stability, stability conditions not predicating the actual study conditions. If they have been kept in bench for 10 hours doing the sample preparation of clinical samples, if they prove it for 8 hours, then there is a lack of stability duration. Similarly, stock solutions and auto sampler stability or freeze stock, keeping inside the freezer and taking it out, how many times they have used, only within the proven stability duration, it has to be sample analysis to be done. If it is exceeds, then again we have to prove for such conditions, and then we have to amend it in the validation data. And partial validations not performed, column lot change or equipment transfer or change in detector or mobile face change. If any changes, if it is there, so partial validation should be done. So in SOP, it should have a provision to do the changes, what are the changes, what experiments is to be conducted, and these details it should be covered in the SOP. And dilution integrity, so clinical samples, if any concentration which comes above the range, linearity range, if it is come, So for that, we have to prove the dilution integrity and we have to do it. But in study, if they got, suppose the linearity higher limit is the 1000 nanogram per ml, we have fixed it, and we got it 1450 like that, and they have proved for 1400 dilution integrity or 1449 like that. If it is there also, it is not relevant. So whatever the concentration which we got in the sample analysis, to that at least one concentration to be higher, only it has to be proved. So if it is 1450 we get, at least we have to prove for 1500 as a dilution integrity to mimic that the concentration after diluting it comes within the linearity. And carryover. So each and every individual runs, we have to inject a blank after the ULFU to show that there is no carryover in each and every individual runs. Carryover doing validation only once if you prove that regulatory may not be accepted. In sample analysis, if you do, so each and every run what we comprises the clinical samples should have a blank followed by ULFU. So each run we have to prove that carryover for that particular run, it is not there. So based upon the instruments, equipments, today it may not come and tomorrow it may come. So it's not from the method, but it can come from the instrument also. So I have done in method validation carryover, but in sample analysis, it is not done. So we cannot expect the data which comes from that is reliable or not. So each and every individual runs, we have to carry over samples also should be present. And next is related to the ISR deficiencies. So ISR not performed as per the regulatory requirement. So they say with the 1,000 samples, first 1,000 samples, we have to do 10%, and more than 1,000 samples, it has to be done as a 5%. So we have to follow that one. And ISR, when we are selecting, 2 time points has to be selected from each of the subjects. So one is near to the Cmax, other at the elimination phase. So how to select it, it is not predefined in the SOP. So that problems is the finding has been came. So we have to predefine in the SOP how randomly we are going to select the ISR samples. ISR failure attributing to the processing error. So without root cause. So when we do the ISR, some failures is happens. So we are not doing any investigations, and without doing it, just we are accepting it. So there the acceptance criteria also they have mentioned. So overall 67% of the samples which we select for the ISR2 should meet less than plus or minus 20% of the original value. If it is not there, then an investigation has to be conducted to identify where the failure happens. Is why the failure happens is it's due to processing error or due to the method robustness or reproducibility. It has to be cross-checked. Selectivity exclusion during ISR, selective data exclusion during ISR investigations. For this also, FDA points they have given. So we cannot exclude this like that. We cannot exclude. So if anything to be investigated, we have to carefully we have to mention in our SOP in which cases the investigations to be taken and how the investigations is to be triggered and how final investigations will be accepted. It has to be defined in the SOP. And ISR results based upon that rejecting of the original run analytical runs. So for this also, some of the CROs they got a 483 is they have got. So ISR result it is coming different, and original result is coming different. And as they identified that ISR it has been failed. So based upon the ISR failure, they go and do the original analytical runs failed, and without any reason, that is not accepted. So if anything, if it is been done, so we have to go for an root cause investigation why ISR failed and why this ISR failure are triggered for the rerun of the original run. It has to be properly defined. So in bioanalytical data report, we should have all the original chromatograms as the repeated chromatograms and re-injected chromatograms. So everything it has to be covered, and no data should be hidden from the regulatory. And this is the fourth one, the preparations and equilibration runs. So usually for a bioanalytical, before running of an runs or before running a system suitability, prep runs will be run to check whether the instrument is been stable or not, or to equilibrate that particular injections, equilibration injections or prep injections will be provided. So some cases what they are doing is they will be running the either QC sample or any of the samples of some clinical samples which they have doubt. So they used to run that particular sample and mentioning as a prep run, they will come to know the ratio and how it behaves like that. Any confident level if they don't have, they used to run and then followed by the run they will be running it. So that it should not be followed. So before system suitability, if you are running any prep runs, that particular concentration should not come from any of your calibration range or from the quality control range. So the concentration selected for using the prep run should not be any of your CCs and QCs. So in such a way, it has to be selected to justify that this particular is a different concentration and we are running the prep injection only for the purpose of equilibrating the system, not to check the processed one, how the samples it is correct or not. So for that, this it has to be followed. And end of run re-injections. Some samples is failed samples is re-injected at the end to say that first QC has been failed. So I have re-injected that QC, second time it has been passed. So like that it should not be done. Re-injections only and what cases the re-injections will be done. It has to be defined in the SOP. So that also it should be due to the instrument failure purposes only re-injections to be done. Due to any acceptance failure of the samples, re-injection should not be performed. And select run re-injections, run re-injected selectively observing CC's QC's failure. So what they will do, they stop the sequence. When it then their CC and QC, if it is failing, stopping the sequences and doing something the adjustments and then re-injecting the entire run and then making it pass. It's not acceptable. So that also it has to be predefined in the SOP. Chromatogram reintegration. So integrating parameters as adjusting and making the passing of the parameters as without any approval from the study director or by switching off the audit trail. If they do such things, is the FDA they have given point. So there the reintegration SOP should be predefined. So without any reintegrations, if it is there, without any study director and QA approval, no reinjection cannot be performed. And if anything reinjection performed after getting the approval, so the original and the reintegrated chromatograms also both should be submitted to the regulatory. So that way we can avoid the FDA finding. And next one is the study director and the QA failures from their view. The FDA findings for study director and this is the protocol did not define evaluation criteria, unless made mental notes and gross observations. So to that avoid, the study director must be a sole scientific authority. Any changes is required, study director has to be signed and it has to be amendment should be done. So protocol, if you identified any criteria to be updated, so it has to be done and as an amendment and it has to be signed by the study director. And software calculations are not validated before this. The CSV has not performed before or using of the systems. So all the equipments should be calibrated and it should be standardized before using for the study. If it is a computerized system with the software, then computer system validation to be performed to ensure the calculations, whatever we get in from the output from the software is right or not. So the CSV to be done for the computer system and then the systems has to be used for the study samples. And final study report based upon the sponsor without notice of the study director, changing of the report is also an FDA point. So any deviations, anything, if it has been there, it has to be documented in the verbatim. Any changes without notice of study director should not be happen in the report also. Coming to SOPs and training, employees are unaware of their roles and deviations not documented in real time. So employees, we have to give must be role specific, training must be given based upon their role. So all SOPs may not applicable for all the persons. So based upon their role, for the activity what they do, for them that particular SOPs training has to be provided, right? And to that, if anything deviated, it has to be get documented. If training record and SOPs are revised, it should be provided. Once after SOP training, without giving training, the involvement of the person for the work is not acceptable. Any SOP trains changes, it has to be trained before involvement of the persons in that activities. And SOP does not specify sample hold time prior to testing. So this is 2025 specific findings for many CROs. They have been given this one. So SOP should have specify the sample hold time. So critical parameters as acceptance criteria for the equipments, for balances, if anything it is there. So how far it has to be there. So the parameters and acceptance criteria, it should be defined in the SOP. And the deviations must be whatever it has, anything if it has been get deviated from the defined procedure or the process. So deviation must be documented contemporaneously. To avoid the FDA findings, this has to be done. And coming to the sample accountability and document change control, for sample accountability, untracked liquids, no records of volumes, how they much used and how the samples came, when it has been taken. So some lack of tracking in the movement of the sample is been there. So complete chain of custody log from receipt to the disposable, it should be available. So from sample receipt from clinical to the end of disposing the track logbook should be available, and freezer temperature expressions log missing or incomplete. So in such cases, continuous freezer temperature data logger and exclusion investigation should be documented. Sample destruction documentation absent. So once we are destructing it, right, we should have a documented procedure and that it has to be provided when they have been asked. Mismatch between the sample log volume and the record analysis. So residual sample written as per protocol should be there. Inter-site sample transfer document, chain of custody form, it should be there. And every places the log should be there. And when we track it or reconstruct it, right, the entire historical things we can able to reconstruct it in such a way in each and every places the logbooks should be provided. And document change control. So chromatographic integration parameter changed without audit trial. So audit trail always it should be enabled. So whenever we do changes, it should be get captured. So both reintegrated and original chromatogram should be there. And the reason should be provided properly. Protocol amendments not approved by the study director. So all amendments it should be approved by the study director, and then only it has to be implemented. The SOP version gaps, supersede version in use on the bench. So SOP already has been revised, but the old version of the SOP, it is there in this one. So for that, we should have a change control procedure, the revision history, and it should be in controlled document. So whenever issuing the revised document, the old documents has to be taken back, and then only the revised version of the control document should be given to the users. In that case, this will be get avoided. Thanks. This is the last. Is there any questions?
Any questions from audience? You can unmute yourself and you can ask. Otherwise, you can post in the chat box. Karthik sir,
Yes, sir.
a great presentation, sir. Thank you so much from our side.
Thank you, sir.
Sir, I have one query.
Yes, sir.
What is the basic difference or the procedure we follow in academics compared to industry? What we lack as far as academics are concerned?
Difference is that when I have been seen from academic and this one, the documentations process and how they have been documenting it, and the software how they will handling for the quantifying the concentrations. So in academics is what they do it, they don't use full-fledged software which is there in that equipment, and they don't calculate it as the software, but it gives the output for the concentration, all those things. I have seen some places they take the values of the response area responses and they put the linearity in the Excel sheet rather than taking it directly from the output which is being given from the software itself. So that it should not be done. So all the quantifications and everything it has to be done. And whereas in academic, they don't follow individual username passwords will not be followed and audit trails will not be enabled. So that we cannot able to identify who has submitted and when it was been submitted, and if anything submitted somebody, if it has been deleted, we will not come to know. So we cannot able to reconstruct the things is when, what, how it has been happened, and if any changes is happened or if anything it has been deleted, we don't know who has been deleted, and we may not able to, once it has been deleted, we cannot able to get back the results. So that lacking it will be there in academic year because they will not much bother about the audit trails. So there loss of data chances are there to lose their data. And academic, they will be having limited number of columns for development trials. So with whatever the columns which they have in their place, so they will be doing the trial with that one only. And what the results they get it, that they'll be using it, and further they may not go as they don't have enough chemistry of columns. They will compromise there itself. So furthermore, our short throughput or better resolutions or better, more sensitive, it cannot be able to achieve because of lack of columns which will be available in the academia. So these are the few things which I have been observed.
Okay, thank you, sir. One question from chat box. Aditya Bogar. How to minimize the enhancement for analyte and internal standard response?
How to increase the enhancement of analyte?
How to minimize the enhancement for analyte and internal standard response?
How to increase the analyte response? So that it depends upon the mass spectrometric optimization. So when we do the optimizations, we have to properly optimize and we have to check because many voltages have been involved in the mass spectrometry, and the pollution energy, exit potential, and nebulizer gases. Few things is also it is there. So when we do the tuning and then optimization, we have to check which particular ion energy or the voltages it's give the more response like that. Initial stage itself we have to do, but once when we do in initial stage, after that we will go for a chromatographic optimization. Once the final chromatographic optimization has been completed, with that mobile phase condition, again we have to go back and do the mass spectrometric optimization. And with that mobile phase condition, and which values is if we change it, we are going to increase the analyte response. We have to see it. And for enhancing the response of IS, as we have to select almost as a regulatory, it has to be go with the isotopic labeled or deuterated internal standards has to be used, because if any loss is there for the analyte, so because if we are going to select a different internal standard, if IS loss is there, it is not going to take care of the analyte loss. So the physiochemical nature of the analyte and the IS, it should be both remain same. If any one, if it has been decreases or increases, both it will be taking care. The final output of the data will not get impact. So internal standards always it should be selected a deuterated or isotopic labeled internal standard, where the internal standards, if you fix it for the analyte, automatically the internal standard response also will be get increased. So there we can able to enhance the responses of the analyte. Otherwise, we have to increase the plasma volume, and we have to check the extraction wise. If we want to see, we have to see how much recovery we are getting it. If low recovery, then obviously the analyte response will be getting less when we compare to the neat or aqueous sample. So to that, already I have been told we have to adjust the for during extraction technique, and we have to select different buffers of trials to be done, and which buffer we are going to get more recovery. So that if you select that, also going to get improve your analyte response, or increasing your aliquot volume, or decreasing the reconstitution volume, and increasing the promoter this injection volume in the LC. These all cases we can able to improve the analyte and internal standard responses.
Yes, sir. One more question from Lalit Sunil Patil. If any patient who is suffering from multiple diseases, then how we target specific target for LC-MS method?
So multiple, we are not going to directly take this particular analyte from the patient sample. We will not be doing. So for the doctor, what is going to prescribe for him the medicine. So that medicine, how it is going to act, for that medicine, how it is going to internally behave in our blood circulations, that we are going to identify this one. But multiple diseases, how we are going to identify. So we are not doing any clinical diagnostics, how it will be done. So we are analyzing, say, how the drug it behaves in our body once we have consumed. So that cases only we are using it for bioanalytical.
One more question from Regal Hariprasad. In industry level, what are the most using extraction techniques?
That I have been told, liquid extractions will be 90% of methods will go with liquid extractions.
Okay, sir.
Hello, sir. My one more question is, how to minimize detector saturation? How to minimize detector saturation without changing the calibration standard concentration?
So detector saturations see that challenges it has been there in most of the companies also. So if it is go with the higher concentrations, the detector saturations chances are more it is there. Still we face some of the molecules detector saturations can be done, but the saturation can be minimized up to 20% rather than the 50% by selecting the column and the right pH, where we can able to avoid the maximum thing. But 100% of detector saturations cannot be achieved. So either we have to reduce the plasma volume what we are going to use, or increase the reconstitution volume to reduce this one. So or by changing the values in the instruments, mass spectrometric values, if we change the voltages, in cases a few up to 30 to 40% improvement can be done, but 100% of detector saturations improvement cannot be avoided. For few molecules there will be there, at least we can able to achieve 80%, we can able to achieve. That remaining 20% for certain molecules, it is not possible to do it. For such cases only, we add an internal standard. So where the response ratios will be taken care, and that 20% will minimize it. We can achieve it there.
Yes, sir. One more question from Pranav Mahajan. How to ensure system suitability and method validation compliance as per regulatory bodies ICH M10 in HPLC based bioanalysis?
So system suitability, we have to run before doing of any individual runs. So system suitability for analytical, one time if you do is okay. But here, before doing any validation activities, experiments, before that, we have to run a system suitability. So system suitability, already I have been told the concentration which you are going to select for the system suitability for bioanalytical, it should not mimic with any of the CCs or QCs concentration. It should be a different concentration from this one, but it should be within the linearity range. So you have to select in such a way the system suitability concentration. 6 replicates injections to be provided, and the 6 injections, once if it has been completed, so we have to check the percentage CV. So the percentage CV should be within plus or minus 15. So 5% it should be there, and the areas also we have to compare. And for that 6 replicate injections, it should be less than 10%. So 10% for the area and 5% for the area ratio, it should be passed. If it is passed only, the validation experiments or any experiments, it can be injected in the instrument.
One more question from Deepika. We do herbal extracts using LCMS.
Herbal extracts.
Yes, sir.
Yes, see, herbal extracts also, it will be having an active ingredient will be there for the herbal. So we have to identify, so which active constituent we are going to identify. If we are able to identify it, that particular molecular weight we can keep it, and for that particular constituent we can able to quantify in the LCMS. If you see some curcumin or some alkaloids, if it is there, so if you know which alkaloids or which thing, if it has been more dominating in that particular extract. So if you are able to identify which alkaloid, that particular alkaloid molecular weight, if it is there, so we can enter that particular mass by charge ratio, and that it can able to quantify it.
Okay, sir. Next, from Sirisha, what are the parameters we have to consider when we go for calculation of regression coefficient?
So parameters.
Yes, sir.
See, we should have at least for bioanalytical, if you see, at least 6 non-zero standards should be there. So and maximum it can be there based upon the range of your linearity, what you are going to fix. Based upon the range, the number of calibration standards will get increased. So about minimum 6 it should be there, and 2 consecutive standards should not be get failed. The lowest, the LLOQ, what we say the lowest standard. So that accuracy, it should be a plus or minus 20%, and remaining all other standards, the accuracy should be plus or minus 15%, and no 2 consecutive standards should fail. That is more than plus or minus 15%, it should not be there. And highest standard should be there, and the lowest standard should also pass to for validation purpose to construct the linearity. And the R square, what we are going to get, it should be more than 0.9850. So if it is more than 0.9850 R square, not R value, we have to consider R square value. So the R square value should be more than 0.9850, it should be there. And the accuracies for all, for LLOQ, plus or minus 20%, remaining other standards should be 15%, and no 2 consecutive should be failed. So these are the requirements for the calibration curve.
One more question
Thank
from
you, sir.
Okay. Yes, ma'am. Go ahead.
Hello, sir. One more question from my side. Actually, the exact flow of the method development of some particular analyte. For example, the first step is a column selection, then second is a mobile face selection, then
No. See,
sample preparation techniques.
whatever the method development, first if you want to initiate it, first we have to see the physiochemical properties, solubility, pKa, log P values, everything it has to be get selected, and then the standards it has to be gone for weighing, and the first it will be for the MS we will be doing it. So MS we have to start tune the parameters. So we have to tune it and we have to check the mass by charge ratio, whether the standard what we got it and same mass by charge we are getting it or not, we have to check. And then with that mass by charge ratio, we have to go for fragmentations. So we have to see the which fragment ion we are going to do it. So if you don't know which fragment ion we are going to select, what are the possible fragments we are getting. So all the possible fragments it has to be selected. And we have to note it down all the instrumentation MS parameters, voltage parameters and gas parameters, everything it has to be noted down. Then we have to go for our mobile phase selection. So in mobile phase, we have to see based upon that what I have been told, the pKa value. So that we have to select organic acetonitrile or methanol, and then buffer which aqueous we are going to use it. Is
But
acidic.
sir, sometimes the PK value it is not working on this stage.
No, if pKa value is that, then we have to go with the trial and error basis we have to do. So we have to check it out. We have to take one organic phase, you keep it at, if it is a quaternary pump, you keep it one acidic base, acidic buffer, another ways you keep it in a basic buffer. So MRM, you with the organic and acidic buffer, how it works out. If it is not been worked out, then you change it with this one. So first you select it whether we'll go with organic, whether with acidic buffer or with the base buffer. So once acid buffer if it has been preferred, then what we have to do, you prepare it with the different pH ratios from low to high, prepare it, and then with each one it has to be injected and we have to check. Okay? So we get the proper selectivity where we are getting it, and proper retention where we get it, and proper peak shape where we will be getting sensitive, how sensitive it is. So we have to check with these the different pHs. We have to check, and if columns it is not coming, then we have to change with the same column. It will be the CA. Different companies are there with a different dimension and with particle sizes. So that it has to be checked it, and then you have to go for which will be work out. If pKa, pH, if nothing we can able to see. So we have to go with a trial and error basis. After that, first MS parameter, then mobile phase column, both it has to be simultaneously we have to check. Once in need, if you achieve standard, lowest and highest standard, if we get the consistency and proportionality, if it is good, then only we have to go for an extraction trial. So first we have to fix mobile column, and then we have to do the extraction technique. If the linearity in neat itself it comes fine, then extraction trial has to be taken.
Okay, Dashari
Okay,
Shyamala.
thank you so much.
Shyamala, do you want to ask any question? Okay,
No,
fine.
sir.
Okay, one more question from Shashank Jaiswal. When selecting a worst case product for cleaning validation, which characteristics are more important?
No, repeat the one more time, sir. Question.
When selecting a worst case product for cleaning validation, which characteristics are more important?
So for cleaning validation, which one it has to be taken?
Yes, sir.
Okay. So cleaning validation, we have to see the drug which is more high concentration levels, because we will be having a lot of standards will be having, and the ranges also it will be get different. So the molecule which the concentration which is more, and if we see that it may not be that much suitable with that particular container. Like if it is been stored in the glass vessels, or if it is been stored in polypropylene tubes, anything it is there. So based upon that, that particular molecule can be selected, and we can do the cleaning validation can be done, or how much exposure we are going to keep it. So any of that particular molecule with high concentration, we think that particular it is going to be kept for a long duration. So in that particular cases, we can take it which molecule with high concentration and exposure to that particular glassware, it is being exposed for longer time. We can take that particular molecule and we can go check it for the cleaning validation.
Okay, sir. Nabi Rasul is asking best sample preparation methods for high throughput studies. You have already answered that. And Manukonda Ritu Medila, how do we select a suitable internal standard when stable isotope labeled compounds are not available?
Now, most of the stable isotopes as well as deuterated, it is there. So if it deuterated and if it is not stable isotopes, if it is not there, so we have to see structurally similar drug it is there or not, and that particular structurally similar drug and the pKa and log P value also, if it is similar nature, it is there. So then that cases it is been accepted, because we also have been done almost for 1 year. We have been trying for one of the internal standard, and we tried it in outside the countries also. And 6 to 7 months we waited and we didn't get it. And finally, we have been selected the structurally similar drug, which is having almost same log P and pKa value. And that we have been done the study, and it has been submitted to the US FDA, and they have came for audit of that particular one, and they asked the reason why it has been not selected. So we have been given our justifications on the documental evidences and mail communications that we have tried a lot and it is not there, but we selected the structurally similar drug which is having the same. We have been used it, and it is also behave in the similar way as that of the drug. So we have given documental evidences to that, and it has been accepted.
So one more question from J. Samnani. How to identify nitrosamine peptides? Nitrosamine peptides and its protein bonds and amino acids using LC-MS.
Nitrosamine is separate, it is there, and the peptides also it is separate. So peptides we have to, because peptides are large molecules, so mass by charge will not be done. So the only the amino acid chain will be selected, and that it will be entered as a mass by charge ratio. And with that chain, it will be around 1,000 plus. It will be there, and the fragment ions also in that particular stable amino acid chain only we will be selecting it, and that it will be used. And for our the technique which we will be using for amino acids is the different where the kits will be available, and the extraction methods itself entirely it will be different. But for large molecules like peptides, the amino acid chain will be taking it one particular chain from this one to this, like that it will be there. So that particular chain, how much Daltons, it is 1,000, 1,100, like that, more than thousands only it will be selected. And similarly, the fragment ions also will be selected from that particular amino acid link from the peptide chains, it will be selected.
Yes, sir. Karthik sir, we have more questions. Shall we proceed?
Better you can.
Yeah, sure, sir. Sure. Yeah. Thank you.
The mail, then we
all your questions will be answered through the WhatsApp group. So kindly bear, resource person is already engaged with some other commitment. Thank you. Over to Karthik sir.
Good afternoon to all. I am Karthikeyan, Assistant Professor from the Department of Pharmaceutical Analysis, JSS College of Pharmacy, Ooty. Sir, it is a great honor for me Sir, it is a great honor for me to stand here and propose the vote of thanks on behalf of the Center of Advanced Analytical and Bioanalytical Science and the Department of Pharmaceutical Analysis. As we come to the end of this very informative and inspiring webinar, we begin by expressing our sincere gratitude to the management of JSS Higher Mysuru for their constant vision and support for their centers. It is because of their quality education and industry connections that events like this are made possible for us. We are deeply thankful to our beloved principal, Dr. S.P. Dhanapal sir, and our respected vice principal, Dr. K.P. Arun sir, JSS College of Pharmacy, Ooty, for always motivating the students and faculty to go beyond the textbooks and learn from the industry. His guidance and support for this webinar helped us in every step of planning and executions. Without his encouragement, the event of this quality could have been the possible. And we sincere thanks the Dr. Krishnaveni, the head of the Department of Pharmaceutical Analysis, for her constant support and guidances. Her leadership keeps our department active and growing, and her encouragement is what inspires us to organize and participate in events like this. When it comes to the most important persons we must thank today is our wonderful research person, Dr. Karthik R, the head of Bioanalytical and Analytical Division, Skydus Pharma Service Private Limited, Chennai. Sir, you made a very difficult subject feel simple and interesting. The way you explained LCMS method and development, bioanalytical validations as the industrial practice and regulatory aspects. It was truly the eye-opening for all of us. Your real-world case studies gave us a real picture of what actually happens in the industry, which no textbooks can teach us. We are very thankful for that you could take time for us, our busy schedule, to share your knowledge and experience with the students, research scholars, and faculty. We will carry this learning with throughout our career. Thank you so much for this, sir. This webinar could not have been happened without the hard work of our coordinators, Dr. B. Babu and Dr. B. Mohan Gandhi, the Associate Professor and Department of Pharmaceutical Analysis. From the planning the event to managing the registration, technical setups, and smooth running of the programs, they handled everything with dedications. We are truly grateful for their efforts. And we also thank all our faculty members of our department who motivated the students to attend and guide us in all the preparations that are present throughout this section. Your support and involvement means a lot to us. And a warm welcome and thank you to all the participants who joined today from the different places, and your active listenings and good questions and made these sessions truly interactive and meaningful. We hope the knowledge and key takeaway from the today webinar will help you in your studies, research, and professional work. As we the end of this session, I feel proud and inspired that knowledge shared today about the LCMS and regulatory science and real industry challenges is something we will remember and use in our future careers. And we look forward to many more such events from the centers and our department. Once again, I thank you to each and every one of you for being the part of this program. Jai Hind.
Karthik sir,
Yes, sir.
thank you so much for your presence, sir. Kindly accept this as token of appreciation from our side.
Thank you so much, sir.
I request all the participants to kindly switch on your cameras for a group photo. Thank you, everyone. Thank you so much, sir.
Thank you, sir.
Thank you, sir.
Thank you so much, sir.
Thank you.